Good morning or good evening, everyone. I hope you are all doing fine. This is the third lecture of histology of the male reproductive system, and in this lecture we will discuss the testicular passages. For the interstitial tissue of the testis, the last time we discussed the testis and its structure. The testis main structure was the seminal vesicle, the seminiferous tubule. This is the seminiferous tubule, okay, and we discussed that this was the Sertoli cell. And these were the spermatogonia. And starting from this cell, this is the primary spermatocyte, and then the sperms are formed. These are the seminiferous tubule. What about the connective tissue in between the seminiferous tubule? This is a seminiferous tubule, okay. Another seminiferous tubule, another one, another one, again here this is a seminiferous tubule, okay. So as you can see here, there are spaces in between the seminiferous tubules. This area, this is a space, it is filled with connective tissue, okay. This connective tissue connect all the seminiferous tubules together. But in this connective tissue as well, you can see you have capillaries. You have capillaries. These capillaries are fenestrated capillaries. Because you have, besides this capillary, you have a cell called interstitial cell of Leydig. Interstitial cells. These cells secrete testosterone, okay. So it is considered an endocrine gland. So it is considered as an endocrine gland. Thus, this testosterone is secreted into the fenestrated capillary. Again, we have here a cell called myoid cell. This is a smooth muscle cell. This is a smooth muscle cell that is present surrounding the seminiferous tubule in order to contract, to allow the sperms to translocate their places, okay. So you have myoid cell, you have interstitial cells, you have fenestrated blood capillaries, and of course you have some fibroblasts. Again, the spaces in between the seminiferous tubules, these spaces are all of these space that I'm labeling, that I'm surrounding, all of this is the interstitial tissue of the testis, and everywhere else in between the seminiferous tubules. Here again, this is the space, this is the space. These cells are the myoid cells, okay, and the interstitial cell of Leydig are pale stained. Any pale stained cell that is present in the interstitium, this is the interstitial cell of Leydig. The interstitial cell of Leydig appear polygonal, where this is one of them, this is another one of them, with the cytoplasm contain lots of vacuolations, pale acidophilic vacuolated cytoplasm, and the nucleus is active and large. The nuclei are active and large. Okay. Okay, and this is the blood capillary, fenestrated capillaries. Okay. For the electron microscope, as you can see, you have in the electron microscopic photo, you have lots of vacuolations. You have lots of vacuolations because it is an androgen secreting cell. You have some mitochondria. Okay. You have extensive smooth endoplasmic reticulum because it secretes testosterone. It also have Golgi apparatus and crystals called Rhynic crystals. Okay, it also have crystals called Rhynic crystals. Okay, and its main function is the secretion of testosterone under the control of interstitial cell stimulating hormone. Okay, here again in the light microscope, the cells appear acidophilic and vacuolated. The nucleus is large and active. Okay, in the electron microscope, the cell contains lots of smooth endoplasmic reticulum and mitochondria. It also contains some Golgi apparatus, lipid droplets, and crystals of Rhynic. The function of the interstitial cell of Leydig is under the control of interstitial cell stimulating hormone, which is the same as LH hormone. Okay. These LH hormones control the interstitial cell of Leydig and its testosterone secretion. Okay. So the FSH hormone affects the Sertoli cell, whereas the interstitial cell of Leydig is affected by LH hormone or interstitial cell stimulating hormone. Whereas for inhibin, it inhibits the FSH secretion in the pituitary and androgen binding protein as well. So inhibin inhibits FSH secretion, so it inhibits androgen binding protein secreted by the Sertoli cells. Okay, now for the ducts, for the duct system. The male genital ducts, there are two types of ducts: intratesticular. Intratesticular means they are inside the tunica albuginea of the testes, and extratesticular, which are outside the tunica albuginea. The ductus efferens is the duct that pass through the tunica albuginea itself. Okay, and goes and start and join together to form the epididymis. So you have three intratesticular ducts, and you have another three extratesticular ducts. For the intratesticular ducts, you have tubuli recti. Okay, this means the straight tubules. Tubuli means tubules. Recti means straight. Retitestis. Retitestis means reticulum. The reticulum that is present inside the testis. And you have, whereas the ductus efferens means the duct that exit. Efferent means exit. The duct that exits the testis. Then Tubuli recti means the duct that is straight, the straight ducts. Reti testis means the reticulum, the part in the testis that part in the ducts that form a reticulum. And ductuli efferens means the duct that goes out or exits the testis. Epididymis, then in the extra-testicular part, you have epididymis, vas deferens, and the urethra itself is considered the last duct in the main genital ducts. Here again, the last part in the seminiferous tubule is called the tubuli recti. The last area in the seminiferous tubule. These are the tubuli recti. The reti testis forms a reticulum in the testis. Okay, this is the reti testis, and the ductus efferens goes outside, pass through the tunica albuginea and goes outside the testis. So the ductus efferens is the last intratesticular duct, whereas the ductus efferens begins to form the epididymis itself. All of this part is considered the epididymis. After the epididymis, you have the vas deferens. Okay, vas deferens. This is the vas deferens. Again, here, inside the testis, they are the intratesticular part. Outside the testis, the first part is called epididymis. Then you have the vas deferens. The vas deferens joined with the seminal vesicle, the duct that comes from the seminal vesicle, to form the ejaculatory duct. Okay, the ejaculatory duct. And this ejaculatory duct join into the urethra inside the prostate, and then the semen or the ejaculate goes into the urethra itself. So again, okay, what was the first intratesticular duct? The first intratesticular duct, what was its name? The first intratesticular duct was the continuation of the seminiferous tubule. This is the seminiferous tubule. The last part of the seminiferous tubule is a straight duct, a straight part. It is called tubuli recti. Then you have a reticulum. This is called reti testis. Then you have a duct that goes outside, exits the testis. It is called the ductus efferens. Then you have the extratesticular duct. The first part of the extratesticular duct is called the epididymis. It is for storage. Then you have the vas deferens. The vas deferens is very long. All of this part is the vas deferens. The last part of the vas deferens join with the seminal vesicle, the duct of the seminal vesicle, and they form the ejaculatory duct. Then this ejaculatory duct joins the urethra in the part of prostatic urethra, and it's the last area of that the ejaculate comes in contact with is the urethra itself, then to outside of the body. Here again, this is a tubuli recti. It is a straight part. It is a continuation. It is a continuation of the seminiferous tubule. And then you have the reti testis, and then you have the ductus efferens. For the tubuli recti, they are present at the apex of each testicular lobule. They are the continuation of the seminiferous tubules. They join to form a straight tubule called the tubuli recti. They are short and straight. Short and straight. Okay. Since they are the continuation of the sertoli of the seminiferous tubule, they are lined by sertoli-like cells. They are lined by sertoli-like cells. Okay. In the first part, whereas they are lined by simple cuboidal epithelium in the last area. Then we have the retitestis. The retitestis, it is the part that form a reticulum. They are the continuation of the straight tubules. They enter into the mediastinum testis, unite to form anastomosing tubules, forming a network. Okay, this retitestis is lined by cuboidal cells, cuboidal epithelium. Okay, and it is surrounded by connective tissue, vascular connective tissue. For the last part of the intratesticular genital glands, you have the ductus efferens. Okay, the ductus efferens. The ductus efferens arise from the upper posterior pool of the retitestis. They are formed of 10 to 20 coiled tubules, and they form the head of testis. Okay, they form head of testis. Oh, sorry, head of epididymis. They form of head of epididymis. Okay, they are lined by simple columnar ciliated epithelium. Okay, the cilia in this part is motile cilia. The cilia in the ductuli efferens are motile cilia. They help the movement of the sperms and the fluid that is present passing in this area. Okay, so it is lined by epithelium, partially ciliated. Some of the cells are ciliated and other are non-ciliated, but the cilia is motile cilia. Okay, surrounding it there is connective tissue, and it is filled with elastic fibers and smooth muscle fibers. Okay, they help in the movement of sperms. The cilia contraction and the muscles that are present both help in the movement of sperm into the epididymis. Okay, for the epididymis, it is a single long, highly convoluted tubules. It is about 6 meters in length. It lies in the posterior aspect of the testis. This part is the epididymis. This is the epididymis. Okay. The epididymis' main function is as a reservoir. This is the epididymis, a single long structure. Okay. The beginning starts from the ductus efferens that are coming from the testis, from the intratesticular duct system, and the epididymis is lined by ciliated cells, but the cilia that are present in the epididymis are non-motile cilia. The cilia in the epididymis are non-motile cilia. Surrounding the epididymis, there is vascular connective tissue, a thin layer of smooth muscle fibers that create peristaltic movements to help in the conduction of sperms to the vas deferens. Okay, so we have smooth muscle fibers present in the ductus efferens, and we have smooth muscle fibers present in the head of epididymis, and around the vas deferens there are also large amount of smooth muscle fibers present. Okay, this is the epididymis. This is a light microscopic picture of the epididymis. The structure that is present in the middle, these are the sperms. These are the sperms. Okay, you can see the epithelium contains cilia. This is the cilia that is present in the epithelium, but this cilia is immotile cilia. Okay, the epithelium is pseudostratified, columnar ciliated, but of course here there is no goblet cells. Okay, so this epithelium is pseudostratified, columnar ciliated, and the cilia are non-motile cilia. Okay, you have to remember this part. The cilia in the epididymis are non-motile cilia. It is called stereocilia, and the stereocilia are just very long microvilli. Okay, surrounding the epididymis you have smooth muscle fibers. If you look closely, you can see some cells that are surrounding each and every tubes. Here you can see another set of cells that are surrounding here. Just simple dots that are present that surround the tube of the epididymis. Okay, so this is the structure of the epididymis. The main function of the epididymis is storage. The main function of the epididymis is storage. While the sperms are stored in the epididymis, it reabsorbs 90% of the fluid. Reabsorb 90% of the fluid leaving the testis. It also secretes a glycerol protein necessary for sperm maturation, so it is very important for sperm maturation. It phagocytoses any residual bodies that are still present inside, that are still attached to the sperm. So it phagocytoses any residual bodies. Okay, and it helps in the capacitation of spermatozoa present in it. This is the capacitation, the beginning of capacitation. Some texts say that it starts in the epididymis, and others say that it starts in the female reproductive system. Capacitation means a series of biochemical changes that occur in the sperms through which they attain their full capacity for fertilization. It is not completed until the sperm, of course, reaches the fallopian tube to fertilize the egg. Okay, so capacitation means the full capacity for fertilization. Capacitation is related to the full capacity of sperm for fertilization, and it is only completed in the female reproductive system, especially in the female fallopian tube. Now for the vas deferens. The vas deferens is a long tube that extends from the tail of the epididymis to the ampulla, where it joins with the duct of the seminal vesicle to form the common ejaculatory duct, which join the urethra in the prostate, join the prostatic urethra. So the vas deferens is a long duct, long muscular tubule, okay, with narrow lumen and thick wall. It starts from the epididymis and ascends within the spermatic cord and passes through the inguinal canal to enter the pelvis. Okay, so it joins something called the spermatic cord. The spermatic cord, we will discuss it after some time. The vas deferens, before it penetrates the prostate, it dilates to form the ampulla of vas, where is connected with the seminal vesicle to form the ejaculatory duct. Okay, the wall of the vas deferens is lined by 3 layers. This is the first duct that is lined by 3 layers. It is lined by mucosa, then musculosa, then adventitia. It is lined by mucosa, musculosa, and adventitia. Okay, the mucosa is the epithelium and the underlying connective tissue. The muscle layer, as you can see here, it is very thick, related to the size of the lumen, the small size of the lumen. And the adventitia is the connective tissue that is present outside the muscle layer. The mucosa, the epithelium, is formed of pseudostratified columnar ciliated epithelium with stereocilia, just like the epididymis that was before it. So it is lined by pseudo-stratified columnar ciliated, cilia are stereocilia. Okay, whereas the muscle layer is divided into 3 parts: inner longitudinal, middle circular, and outer longitudinal muscle fibers, and then it is covered by loose connective tissue forming the adventitia. The vas deferens form powerful peristaltic contractions using the smooth muscles during ejaculation to convey the sperms into the ejaculatory duct. Okay, the main function of the duct deferens is to rapidly transfer the sperms from the epididymis to the ejaculatory duct to the urethra. Therefore, it contains very thick muscle layer. This is a section in the vas deferens showing the mucosa, which is pseudostratified columnar epithelium with stereocilia. It is lined by stereocilia, and the lamina propria is just some connective tissue underneath this epithelium. It has a thick wall formed of smooth muscle fibers and some collagen fibers. This bluish stain stains the collagen fibers. As you can see in the outside part here, this part contains lots of collagen fibers. It is adventitia, or some connective tissue in between blood vessels. This area contains lots and lots of collagen fibers. This part is the lamina propria of the mucosa, and in between the pale area are the smooth muscle fibers, and the darkened area, the bluish areas, are the collagen fibers. Whereas in the other photo, this is H&E. The pale part here, the pinkish part, is the lamina propria, and all of these reddish dots, reddish structures that form circles surrounding the lumen, these circles, these are the smooth muscle fibers, and then the outside part, which is the pale, is the collagen fiber. So in this slide, blue means collagen, means collagen, okay, whereas in the other one, red, the more acidophilia means muscle fibers, whereas the less acidophilic parts are the collagen fibers. Okay, the main function of the vas deferens is powerful peristaltic contraction of smooth muscles during ejaculation to convey spermatozoa to the ejaculatory duct. Okay, so this is the mucosa of the vas deferens. You have three muscle layers, inner and outer longitudinal, whereas the middle is circular, smooth muscle fibers, and the outermost part is adventitia. The vas deferens is a very long tube that sends the ejaculate from the epididymis, transfers the ejaculate from the epididymis up to the urethra via the ejaculatory duct. For the ejaculatory duct, it is formed of the union of the ampulla of vas deferens with the duct of the seminal vesicle. This is the seminal vesicle. This is the duct, the ampulla of vas, the joining of both of them. So let's say this is the vas, and the vas form a dilatation at the end. Okay, this is the vas, and this is the seminal vesicle, its ducts. When both of them join together, they form the ejaculatory duct. So this part is the ejaculatory duct, and this part enters the prostate to join the urethra. So this is the urethra, and this is the ejaculatory duct. This is the ampulla of vas, and this is the seminal vesicle. Okay. It is lined by simple columnar epithelium and is surrounded by connective tissue, but there is no muscle in the wall. Okay, there is no muscle in the wall. The duct empties its content into the prostatic urethra. What is the difference between the vas deferens and the ureter? The vas deferens is part of the extra testicular ducts, whereas the ureter is part of the renal system. Okay, urinary system. Okay, both of them are very narrow. Okay, they may appear star shape, the lumen may appear star shape. Okay, but the main difference is the epithelium lining. The epithelium lining, the vas deferens, is pseudostratified columnar with stereocilia, whereas the epithelium lining the ureter is transitional epithelium. If you can remember, the transitional epithelium was stratified epithelium with multiple layers of cells, and the last cell was called a facet cell. The last cell on the surface was a cuboidal cell, maybe uninucleated or binucleated. Okay, whereas in the vas deferens, the cells are pseudostratified columnar with stereocilia on the surface, Okay. Okay, so the vas deferens is lined by single line of cells, whereas the ureter is lined by multiple layers of cells. The vas deferens has a narrow layer of loose connective tissue, which is lamina propria, whereas in the ureter the lamina propria is a little bit wider. Okay, the muscle layer in the vas deferens is very thick. The vas deferens is very thick and well defined, whereas the muscle layer in the ureter is much thinner and less developed. Both contain inner longitudinal thick middle circular and outer longitudinal smooth muscle layers. Okay, but again in the vas deferens the musculosa is very thick, where in the ureter the musculosa is much thinner. The covering layer is the adventitia, or the surrounding epithelium. The surrounding structures are spermatic cord structures, spermatic fascia. Okay, whereas in the ureter it is partially covered by peritoneum. Partially covered by peritoneum. There is no other structure besides the ureter, whereas in the vas deferens, as you will see in the next couple of slides, you will find that you have the spermatic, the vas deferens, and lots of veins and artery, and you have skeletal muscles that are present, and this structure is called the spermatic cord, whereas in the ureter it is partially covered by peritoneum. So as you can see here, this is the vas deferens. This part of the slide is the vas, whereas this part of the slide is the ureter. The vas deferens, the lumen is narrow. The ureter the lumen is a little bit wider. The epithelium of the ureter is transitional epithelium. As you can see, this is the facet cells that are present on the surface. The cell may be uninucleated or binucleated, okay, and they have special characters. Whereas in the vas, the epithelium is pseudostratified columnar. You have cell on the surface and cell underneath, cell on the surface and then underneath, okay, and you have stereocilia that is present. So here the epithelium is pseudo stratified columnar ciliated with stereocilia. Okay, the muscle layer in both structures is inner or outer longitudinal and middle circular, but the main difference is the thickness. The thickness of the muscle in the vas deferens is much larger related to the lumen, whereas the thickness of the wall in the ureter is thinner, much thinner than the vas deferens. The spermatic cord is formed of 4 structures. The first structure is the vas deferens, okay. The second structure is the pampiniform plexus of veins, The third structure is the testicular artery and vein, and you have the cremasteric muscles, and of course you have lots of nerves, nerve supply present, okay. So the main 4 structures are vas deferens, pampiniform plexus of veins, testicular artery and vein, and cremasteric muscle. Cremasteric muscle are striated muscle fibers, okay. The pampiniform plexus of vein, its main function is to release heat, so that the temperature in the testis would decrease by 3 to 4 degrees Celsius than the body. to help in spermiogenesis. Okay, the vas deferens, its main function is transportation. Okay, this is the spermatic cord. The spermatic cord is formed of vas deferens, the lumen of the vas deferens. What was the lining of the mucosa, the epithelium that lines the mucosa? What was its type? Was it simple or stratified epithelium? It was simple. What type? Pseudostratified, columnar ciliated with stereocilia. The cilia are stereocilia. And you have the muscle layer. The muscle layer was formed of 3 parts. What were the 3 parts? Inner, middle, and outer. The inner was what? Inner was longitudinal, and the outer was longitudinal smooth muscle fibers, and the middle was circular smooth muscle fibers. Okay, and then you have a little loose connective tissue present, which is the adventitia. The surrounding connective tissue is the spermatic cord for the vas. You have the testicular artery and vein. Testicular artery and vein. This is the artery, and this is a vein. You have a plexus of veins, pampiniform plexus of veins, and you have cremasteric muscles that is not seen in this photo. Okay, then you have 4 structures in the spermatic cord. You have vas deferens, you have testicular artery and vein, you have pampiniform plexus of veins, and you have the cremasteric muscles. These are the different lining of the different parts of the testicular ducts. You have intratesticular duct and you have extratesticular duct. The first 3 parts, these first part, these are the intratesticular, whereas the lower part is the extratesticular. The intratesticular, the first structure is the seminiferous tubule, which is the site of spermiogenesis, and then you have tubuli recti, okay, the straight tubules. The tubuli recti was lined by what? It was the first part, it was dividing into 2 halves. The first half is lined by sertoli, like cells. And the second part was lined by what? The second half was lined by simple cuboidal epithelium. Okay, and there was no smooth muscle fiber surrounding the retitestis by any means. Okay, but the tubuli recti by any means. Then you have the retitestis. The retitestis form a reticulum in the posterior pool of the testis. What was the lining epithelium of the retitestis? It was as the previous part of the straight tubules. It is lined by simple cuboidal or cubical whatsoever, cuboidal epithelium. Okay, for the ductus ductuli efferens, it is the exit. Efferent means exit. It exits the testis. Okay, it exits the testis, and it is lined by what was the ductus efferens lined by? It was lined by simple columnar epithelium with cilia. Some was ciliated and some was non-ciliated. Ciliated and some was non-ciliated. Some cells. Okay, the cilia here was motile cilia. The cilia was motile cilia. Okay, so as you can see, The seminiferous tubule had Sertoli cell and the tubular recti had Sertoli cell. The tubular recti then had cuboidal epithelium and the retitestis had cuboidal epithelium. Then the epithelium enlarged a little bit, forming columnar epithelium. And in the epididymis, the columnar epithelium that was ciliated here, it changed into pseudostratified columnar ciliated epithelium, but the change would be the cilia is stereocilia. The muscle layers start from the dactyli efferents, or it starts from the dactyli efferents and persists till the ejaculatory duct. In the ejaculatory duct there is no muscle layers. Okay, for the vas deferens, the epithelium is the same as the epididymis. Okay, whereas in the ejaculatory duct, since the ejaculatory duct is the junction between the vas deferens and the duct of the seminal vesicle, so the epithelium changes into simple columnar epithelium. And then in the urethra, according to the site of the urethra, the first part of the urethra is in the prostatic urethra. It is lined by transitional epithelium, same as the urinary bladder itself. Then you have the membranous urethra. The membranous urethra is lined by stratified columnar or pseudo-stratified columnar epithelium. Then the penile urethra is lined by stratified columnar epithelium. And in the last part of the male urethra, the fossa navicularis, it is lined by stratified squamous non-keratinized epithelium, since it is the last part that would change into skin afterwards. Okay, so again, the lining epithelium of different parts of in which the sperm pass. Okay, the first is the seminiferous tubule. It is lined by spermatogenic cells and Sertoli cell. Then you have the tubuli recti. It is lined by Sertoli-like cells, same as previous Sertoli-like cells, and then simple columnar epithelium. The retitestis is lined by, same as before, same as the part that was before it. It is lined by simple cuboidal epithelium. Then you have the dactyli efferent. It is lined by simple columnar epithelium, either ciliated and non-ciliated cells. The cilia here is motile. All these structures, the tubular recti and the retitestis, have no smooth muscle fibers. Actually, the seminiferous tubule contain myoid cells, surrounded by myoid cells, which are a variant of smooth muscle fibers. Then you have the ductus epididymis. It is lined by pseudostratified columnar ciliated epithelium. Then the vas deferens lined by the same type of epithelium. The ejaculatory duct, since it is a junction between two structures, then it is lined by simple columnar epithelium. And the last part, the urethra, it is lined by, according to its site, the prostatic urethra lined by transitional epithelium. Then the stratified columnar and pseudostratified columnar epithelium are the lining of membranous urethra, and the penile urethra lined by stratified columnar epithelium. And lastly, the fossa navicularis is lined by stratified squamous non-keratinized epithelium. The structures that have smooth muscle fiber, true smooth muscle fiber surrounding, are the ductus efferent, the epididymis, and the vas deferens. Thank you, and I hope that you learned the different parts of the male genital duct, either intra or extra testicular parts, and hope to see that you listen to the next lecture, inshallah.